and since that time I know her, and she has finished her PhD from TIFR in 2011 and then spent about 3 years at Brandeis University doing postdoc. She is very bright and a very creative experimentalist. She works on soft matter, and looks at the systems from an experimental point of view. And also she actively collaborates with theorists. So this is for the students. Just, uh, Prerna, you won't mind taking questions in the middle of the talk, right?
Yeah. It's okay. I'm not in a hurry to cover anything. We
Yeah.
can just.
So this is especially for the students. Please ask questions and feel free to ask as many questions as you want.
Yeah.
So with that, I would like to ask Prerna to start the talk.
Sure. Thank you, Shakuntala. So, share my screen. Okay, just did that. Screen share. Okay, so welcome to this seminar on mechanics of ciliary beating and phototaxis. It's a great pleasure to give a talk at the S.N. Bose Center for Basic Science. I'm at the Department of Physics, IISc Bangalore. This work was funded by India Alliance, DBT Welcome, and of course my host institute, IISc. So today I'm going to talk to you about this organelle called Cilia. And is there a pointer that I could use? Sorry, pointer options. Okay. So I'm going to talk to you about this organelle called Cilia. It's a filament-shaped object that sits on our the epithelial tissue of our lungs, and its role is to oscillate in a mucus-like environment and push the debris out of our lungs. So I'm going to talk to you about its the forces that act on the cilia and the role of the fluid surrounding it and so on. And if time permits, I will also discuss this behavior called phototaxis. So in addition to human beings who use cilia for for having clean lungs, the same filament is used by microorganisms to swim in fluids. And a fundamental behavior of these microorganisms is that they run towards light. So phototaxis literally means directed motion towards a light. And I'll try to elude some interesting things about these phenomena. So first I'll acknowledge my co-workers on these projects. So Debasmita and Ronojoy were involved in the Cilia work, and Sujit and Aparna were involved in the phototaxis work. So the outline of my talk is I'm going to first show you a general class of filaments, which are called active filaments, and motivate the phenomena of ciliary beating in the sense that cilia is a class, cilia is an example of an active filament. And then I'm going to highlight some experiments that have looked at the role of hydrodynamics in ciliary beating, and then also the role of internal friction in biopolymers. And with these experiments in our radar, I will then showcase our recent experiments on looking at the beating waveform and flow field of an active cilium. And then the part 2 of the same talk will look at the collective phototaxis, and then I'll conclude. So active filaments are essentially chain-like systems that are out of equilibrium and undergo spontaneous oscillations by converting chemical or other forms of energy into mechanical motion. So here you're looking at a chain of superparamagnetic colloidal particles, and I'll play the movie, and what it will show is that this chain would go back and forth because there is a time varying oscillating magnetic field. And strictly speaking, it's not a system that is undergoing spontaneous oscillations, rather it's a driven system. And there are other examples where um, people have looked at what are called Janus colloids, which are half metallic and half dielectric. And so each of this sphere is half metallic and half dielectric. And these spheres undergo some kind of attractive interactions and form a chain. And now if you put this chain in AC electric field perpendicular to the imaging plane, what happens is that there are induced electroosmotic flows on the surface of these spheres, which are asymmetric in the sense that the metallic side has more flow than the dielectric side. And that leads to self-propulsion of these particles. And I'll play the movie, and what you will see is that this chain does these beautiful bends and goes back and forth in a very elegant manner. And what you will notice is that all the spheres are always moving with their dark side in the front, which is the dielectric side. So of course this is a very complex phenomena, and I don't intend to show you all the features of it. And then there have been also simulations where the authors are interested in, for example, looking at chains of beads, which are or beads or spheres, which are polar particles and self-propelled. And then these active stresses lead to buckling, spontaneous buckling of the chain. So all of these examples actually are inspired by the biological phenomena of ciliary beating. So let me show you what this beating looks like in the biological context. And it really is the basis of our lives, right from our existence to our growth to our survival. And here I'm showing you the example of survival in the sense that you're looking at a mouse airway lungs, so the epithelial tissue of the lungs. And I'll play the movie. You'll see these hair-like objects that are going back and forth. And this motion sets up fluid flow on top of this epithelial tissue. And if you put, for example, tracer particles, as this study did, you will see that the tracers unidirectionally move. So this is showcasing how the debris in our lungs is actually transported across the tissue. And you can see that this is like thousands of ciliated cells, each of them carrying hundreds of cilia that are beating and producing this macroscopic fluid flow around them. As I said in the beginning of my talk, that cilia is not just used by higher order organisms, it's also used by simpler microorganisms. So here you are looking at a single celled organism called paramecium. It's about 50 microns in size, and its surface is coated with hundreds of cilia. And this organism essentially swims because the cilia exerts force on the fluid, and then this fluid motion then propels the microorganism forward, right? And you can see that there is like long wavelength modulation of the phase of the beating across this organism. You can think about even simpler organisms like sperms, which have a single cilia associated with them. So this is what I meant by cilia deciding our existence, because it is through the beating of the cilia that the sperm actually reaches the ovum and fertilizes it. And here I'm going to play the movie, and you can see that through whipping this organelle, the sperm moves forward. And then the third microorganism that I would like to draw your attention to is Chlamydomonas Renharti. It's a single-celled algae which has two cilia on its head, and so all of these microorganisms are essentially using ciliary beating to propel themselves through the fluid. And out of these three, Chlamydomonas is a workhorse in my laboratory and my favorite organism, because you can grow it in really large quantities. So experimentally, you can get enough material to work with. And there is a library of mutants that one can explore that are available in an open source manner. And from an technology point of view, also these organisms, the Chlamydomonas is an important organism because it's being explored for biofuel production, and it's routinely used in bioreactors for all kinds of bioengineering purposes. And so, and it's a model organism in biology for things like DNA damage, metabolism, and so on. So, with this significance of the organism, I'll show you that how these cilia beat on Chlamydomonas. So here I'm holding the Chlamydomonas cell on a micropipette, and the flagella are beating in a beautiful synchronized manner. And if the cell was not held on the micropipette, the cell would actually swim. Now, even though I started off this talk by motivating about uh, transport in lung tissue and so on, you may wonder that whether it makes sense to study cilia from Chlamydomonas. And it does make sense because structurally the organelle is highly conserved. That means the cilia that is present in Chlamydomonas Renharti has very similar structure compared to the cilia that would be present on our lungs. And the structure of this organelle is very unique, and leads to the complex function that we see. So it is made up of 9 microtubule doublets that are cross-linked to each other through these dynein arms. And so dynein is a molecular motor whose role is to slide the doublets with respect to each other. Okay, and then there are cross-linking proteins which are called nexin links present also within the structure. So the way this filament actually beats is that the dynein motors slide microtubule doublets, but the doublets are cross-linked with these passive linker proteins called nexin links. And so essentially this sliding is converted into bending, because you have when you slide, these cross links provide a component of the a normal force to the filament and bends it. This system is a low Reynolds number because the length of the filament is about 10 microns and the diameter is about 200 nanometers. And the filament beats at about 50 hertz. So if you plug in the numbers for the Reynolds number, it is significantly below 1, and where you can say that the inertia is irrelevant and friction should dominate. The molecular motors slide the microtubules by consuming energy in the form of ATP. So ATP is like the fuel that these motors use to perform work. And so it's a continuously driven system in the sense that within the organism, the motors are continuously consuming energy and doing work on the filament. And therefore, the filament will be in a dynamical steady state only when the elastic stresses due to the drive are balanced by some form of dissipation, right? So the dissipation has to dominate, otherwise the system will not be will basically going into some kind of runaway situation. So usually ciliary beating, or the standard way to analyze ciliary beating is to involve only fluid friction as the source of dissipation, and also to measure only the waveform of the beating. So for example, typically what is done is that you measure the waveform of this filament and you write down an equation of motion which balances the active moment due to the molecular motors with the passive stresses of the system. And then you have to use different constitutive models for the active moment, because the passive stresses are in the terms of macroscopic variables like the curvature of the filament, right? But how does the active force due to the molecular motors depend on things like curvature is not known a priori. So you have to use a constitutive model for active moment in terms of these macroscopic variables and solve for the waveform. And when you do that, you can you will have to use some kind of parameter optimization to get best fit. And here, for example, the authors found out in a certain kind of model of active moment that the fundamental mode of shear angle along the filament can be well modeled, can be fit with this sort of approach. So this is the introduction part of my talk, where I have told you what ciliary beating is and how typically a physics person would analyze it. And now I'm going to show you some experiments that probe the role of fluid friction in ciliary beating.
Prerna, I have a question.
Yes, yes.
You had shown that the synchronous beating of two cilia for
Yes.
DC Renharti,
Yes.
which actually takes it forward.
Right.
Now, how do they change direction? Is it for the beating becomes asynchronous or something like that?
Yeah. So that's a very good question. So the organism turns by changing the frequency of one of the flagella. So you slow down one flagella relative to the other, and then that allows the organism to turn.
Okay.
So when you have a frequency difference between the two flagella, you technically lose synchronization in that sense. Yeah.
Okay. Thanks.
Right. Thanks. So
Prerna, there is another question.
yes.
Abhinav, you can ask directly.
Yeah.
So I didn't quite understand what is like the passive forces are exerted by what, the fluid, or is it because there's the something inside cilia only which is exerting these forces?
Right. So there are two kinds of passive stresses. One is, you have the fluid friction, right?
Right.
And then the other is that the filament is an elastic filament. In the first approximation, you would think of it like an elastic beam. So when the motors apply these moments or torques and bend the filament, then you have a elastic restoring force.
Oh, okay. Yes.
That is built in the filament. Yeah.
Okay.
I can't see who needs to ask questions, so Shakuntala has to moderate that for me. Yeah.
Yes, I will do that. I will do that.
Thanks. So, so now I'll show you some experiments on how. So the role of fluid friction in ciliary beating in context of Chlamydomonas is highly debated, and I'll showcase a few of those experiments and tell you where the intricacies lie. So the role of fluid friction has been extensively studied in the context of synchronization of these two flagella on chlamydia. I will be using the terms flagella and cilia interchangeably. So here in this experiment, the authors held the cell on a glass needle and then used a piezoelectric transducer to move the container in a periodic manner. So the cell is fixed and the surrounding container is oscillating periodically. And then the authors asked that for what parameters of this periodic movement there will be a phase locking between this external flow and the flagella of Chlamydomonas. And what they found out was that it was possible to get the flagellar beating to follow the motion of the container, but the magnitude of the hydrodynamic forces that were required for the synchronization were much larger than what the organism would encounter in its natural habitat or natural free fluid swimming. And so they concluded that there must be a synchronization mechanism that is internal to the cell. And they proposed that what is happening is that the two cilia are actually elastically coupled to each other within the cell. So within the cell, they are embedded in what is called a basal body. And then the two flagella are connected through these distal fibers. Now, so essentially, because of this coupling, elastic coupling, the authors concluded that the synchronization is evident. And if you make mutants where you disrupt these distal fibers, then the organism loses the synchronized beating. And
Prerna, I have a
yes,
question.
yes, yes,
So I'm just trying to
right,
understand the aim of the experiment. Here,
right,
the authors were looking for at which oscillation frequency of the piezo
right.
they find the synchronous beating of the two cilia. Is that right?
Uh, so they basically they wanted to know because they're imposing an external fluid drag on the organism, right? So
Right.
this periodic beating frequency times amplitude will give you some kind of fluid power. And what they were looking for is that uh, for example, when this container moves backward, then do the flagella move backward with it, and then when the container comes forward, do the flagella follow it.
So they are
And
not looking at the presence of synchronization between the two flagella. That is
yeah,
not
yeah,
their
yeah,
concern.
yeah. I mean, so it was some kind of how should I say?
Is
Yeah,
that
so it is like you have a harmonic oscillator, and then you are asking whether you can get it to beat at the frequency that you have imposed.
So like a
You know.
resonance.
Yeah. So, I mean, they were not looking for resonance, but like in principle, yes, you would hit that. Yeah.
so? Just curious. In
Yeah.
this setup, suppose I use a very slow oscillation frequency of the
Yes.
piezo.
Yes.
Then, and if I if I look at the two flagella,
Yeah.
then do I find how do they behave? Do they beat synchronously?
Yeah, so for very slow flows, the flagella will not really care about the drag is too low to actually influence it. So
So then, what do? They will be independent of each other.
no. So see, the thing is that okay. So the okay. So the the organism has long time periods where flagella synchronized, and then there is noise in the system which makes it lose synchrony for some time, which are called phase slips, and then it picks up synchrony again. So this is in an ambient quiescent fluid. Now when you impose this external fluid flow, then the question the authors were trying to ask is at what at what fluid drag will the this, even though the flagella will be synchronized with each other, will they also synchronize with the
The
movement
applied
of the cell?
frequency. I see. Okay.
Yeah.
Okay.
Okay. And the reason they were trying to do that is because the idea was that in the quiescent fluid, let's say one flagella is moving and it's setting up a fluid flow which then influences the other flagella, right? And so that is what they thought of as a fluid coupling leading to synchronization. So in an ambient fluid you can't probe it, right? Because this is like a live organism, and it's hard to establish flow field based couplings that way. So that is why they were doing this active, like a driven experiment. So
Okay.
yeah, so that is why this conclusion was important, that they could synchronize the flagella to the motion of the stage, but at very large forces of hydrodynamic or hydrodynamic dissipation or forces. So I mean, their conclusion was that the fluid flow based coupling between the two flagella is not possible, because in order to synchronize with this imposed flow, the forces that they required were much higher than what the organism would encounter naturally. I hope this is
Okay, okay.
clear.
Joydev, you have a question?
No, it's okay. No.
Okay. So, yeah. Then the the next study that followed up on this study was to impose a steady flow on the organism. So instead of asking whether the flagella synchronize with the imposed flow, they just imposed a steady flow against the organism, and then asked how do the beat parameters change, how does the amplitude and how does the phase speed change in response to imposed flow of speed u. So these curves are showing rate of change of amplitude with the flow velocity and rate of change of phase speed with the flow velocity across the beating waveform. And by writing down simple equations of motion, what they realized was that they could not fit the data at all unless they introduce an extra source of friction, which they hypothesized to be the internal friction. It's a friction that is present, whose source is present within the cilium itself. And essentially, this study uh, assumed that the fluid friction obeys what is called resistive force theory. I will come to it in the next slide. And modeled internal friction as a free parameter that was effectively a renormalized fluid friction. So their point was that in order to fit the experimental data, they needed to artificially increase the viscosity of the ambient fluid by a factor of 5. So they said, okay, there must be some other source of dissipation that's present inside the filament. So there were a number of things that were left to be explored, and a lot of things were sort of assumed. For example, this resistive force theory and so on. So what we really wanted to do was to explore, uh, to uh, to have a unambiguous experiment which shows that there is whether there is internal friction present in ciliary beating or not. So before I tell you about the experiments that we did, I also wanted to tell you the background of this internal friction literature. So internal friction has been postulated for many biopolymers and polymers in general, and I'll showcase some of these experiments. And so the source of internal friction is typically ascribed to some slow structural rearrangements, or essentially saying that your object has internal degrees of freedom that are dissipated. So in this study, the authors were looking at thermal fluctuations of microtubules. So microtubules are building blocks of cilia, right? And these are very rigid objects, and you are just observing their contours through Brownian motion, through thermal fluctuations. And they measured the relaxation time of bending normal modes versus their wave vector. So for something like actin, which is another standard biopolymer in our bodies, in our cells, actin showed a 1 over q to the power 4 scaling, which is expected from classical elasticity. But microtubules showed that above a critical wave vector, the relaxation time was becoming independent of the wave vector. the data is of course noisy, and but at the same time it's not something that you can ignore. So the way this data was explained was in this way. So for actin, you will write down an equation of motion. So this is basically force per unit length that is acting on the filament. U is the transverse displacement of the filament. B is the bending modulus. So the first term is the elastic restoring force. Essentially, this term comes from the fact that the bending moment is proportional to the curvature of the filament. The second term is the dissipation due to the surrounding fluid. And the third term is the thermal force delta correlated average zero and so on, the standard thermal force. And from here you can see that the relaxation time of the bending modes should scale as eta by bq to the power 4 because of this.
Sorry, one
Yes.
quick question.
Yes.
Here I see you have not taken into account the surface tension term, just the bending rigidity.
Yes.
So what is the reason for that?
Basically,
You expect it is too small a
yeah.
contribution? Okay.
Right. I mean, these are like, you can think of them literally like solid objects suspended in water. I mean,
Okay.
yeah, and completely immersed in water, right? So, yeah.
Okay.
It's a good question in the sense that one could have these proteins on surfaces of these polymers, which could lead to slip and so on. But because we are doing a coarse grain modeling, we are ignoring that in that sense. And then the data for microtubules, of course, doesn't obey this 1 over q to the power 4 scaling. And the way this was resolved is to add another term by hand and call it the contribution from the internal friction. So what is this term doing? So what this term is really doing is it's saying that it depends on the time derivative of del 4u del s4. So the way it is stemming, or the origin of this term is in the fact that now you are saying that your bending moment not only depends on the absolute value of the curvature, it also depends on the time rate of curvature, or rate of change of temporal rate of change of curvature. And then you have this coefficient, which is the internal viscosity. R is the radius of cross section, or the radius of the polymer. So this r to the power 4 is set so that you can your coefficient has the correct units of viscosity. And now your relaxation time will scale in this way. And you can see that the internal friction term will give you a wavelength independent contribution, because q to the power 4 will cancel out q to the power 4. So this was one of the earlier studies which started to talk about internal friction in biopolymers. And another source of friction that is relevant for cilia, or internal friction that is relevant for cilia, is the friction due to motors themselves. So these motors actually walk on the filament, and through this walking they tend to slide the microtubules. So now in this experiment, actually the motor is so this experiment was conducted in absence of the fuel, so the motor is not really walking on the track. So the track is the microtubule. So microtubule is fixed on the surface, so it's immobile. And then you have a colloidal particle which has this motor protein attached to it. And so this motor protein is interacting with this microtubule. And now what you do is that you oscillate this cover slip back and forth with some velocity v. And when you move the microtubule, this motor protein, since it is interacting with the microtubule, it will also get dragged with it, and therefore it will drag the microsphere away from the center of the trap. And so you can measure the force that is acting on the microsphere. And if you plot that friction force as a function of speed, what you find is that instead of getting a straight line, you get this non-linear curves. And these non-linear curves are essentially saying that at high enough pulling speed, there is bond rupture that happens between the track and the microtubule and the motor protein. So when there is a bond rupture or dissipative forces that act, then this motor kind of slips off, and the the bead doesn't move as much as it would have moved if you were in a pure hydrodynamic regime. So these are the sources of friction in the building blocks of cilia. Now, there were these classic experiments which were trying to measure the elasticity of cilia. So here you have immobilized cilia on a cover slip, and you take a glass needle and you apply a force on this needle at time t equal to 0, and you measure the displacement of the tip of this needle as a function of time. So think of it like a constant stress experiment. And so the strain builds up, it builds up slowly over time, and then also it doesn't settle, it kind of continues to have a creep like flow. So these kinds of plots tell you that it's not a good idea to model cilia as a pure elastic object. It has these sources of internal friction within them. Now, internal friction is not just relevant to these biopolymers that I've shown so far. It's also being talked about in the context of protein folding. And so, in this series of experiments, the authors were measuring. So basically, they were adding different concentrations of denaturant. So proteins are usually folded into some kind of well-defined structure. And if you add a denaturant, uh, the chain sort of opens up, right? And then, given enough time, the chain would slowly try to refold back. And so they were measuring these refolding time scales as a function of the viscosity of the solvent. So now what they found out was that for low denaturation, the extrapolation was not going to zero, meaning at low and low viscosity, the refolding time was not going to zero. And so that again tells you that uh, essentially what's happening is that the protein has these internal degrees of freedom. The chain can rotate around itself, can wrap around itself. There is things like reptation, and all of these lead to sort of an energy barrier for the protein to go to its equilibrium state. So, in addition to the viscosity of the fluid, which decides the time scales over which the protein can explore many different configurations and minimize its energy, there are these internal degrees of freedom within these proteins that also lead to a, a finite time scale that's required for the protein to fold. So this is sort of the background of my study. And now in the last 20 minutes, I will try to show you what we did and how we wanted to approach this problem and remove some of the ambiguities that were left unattended in the previous set of experiments by other authors. And so the system that we wanted to study are called reactivated axons. So here is again a Chlamydomonas cell which has two flagella. So I think by now you can appreciate these experiments become very complicated because first of all you have two flagella that can interact with each other, and then they are part of a live cell who also can in principle respond to fluid stresses and other kinds of stresses that it may encounter. And therefore, to really know the mechanics of a single filament and the role of fluid around it, uh, it's very hard. And so it's hard in terms of deciphering which when the data, so which part of the data is originating from which part of this complex system. So we decided to actually remove these flagella from the cell. So when we give certain kind of drugs to the cell, it's a natural tendency for this organism to shed its flagella, and that flagella is completely brownian as shown in this movie. The reason it sheds this is because this organism has the ability to regrow its flagella. So this is a prey predator response in the sense that if clammy feels that a predator is holding its cilia or flagella, it will actually shed it and try to get away from the predator in that way. So here we are with some of these, Brownian objects. Now remember, the object is 9 microtubule doublets with molecular motors all around them. So the motors are still there. If I add the source of energy, which is ATP, the filament should beat, and it actually does. So here is the same filament, and I have added ATP, and it is freely swimming within the fluid. This is free swimming. We can anchor this flagella on a cover slip and watch it beat. Okay. And so,
Excuse me.
yes.
So was this all done experimentally, or is it in a simulation that like we are imposing conditions?
No, this is
Oh,
experiment.
that is really,
It's like chopping somebody's arm and getting it to do work.
yeah, like it really feels good. Like, okay, so amazing that we can add ATP and then
Yes.
have it
Yes.
do the work
Yes.
without requiring the whole body to,
Yes.
you know,
It
yeah,
is one of the
that's,
most amazing experiments I have done.
yeah, it's really
So,
amazing.
yeah.
And Prerna, for this
Yes.
isolated flagella,
Yes.
you will not expect existence of internal friction, will you?
Yes, we will, because all the proteins are intact. So there is the reason I expect it is because of following. The building block of this filament is still microtubules, which by themselves have shown whose bending
Okay.
fluctuations showed internal friction. And then I also showed you experiments where the motor friction was also dissipated.
Okay.
And so, yeah. in
So
that
those
case-
factors are still there.
Yeah,
Okay.
yeah.
Thanks.
So that is why I showed those experiments to motivate that why we would expect it in this filament. And so these are simple boundary conditions that I can impose as an experimentalist, and they're free from the control by cell body, and I'm looking at the mechanochemistry of the motors alone. So now what we did, which is different from most of the studies before us. So this purifying flagella and reactivating flagella, even though it looks really beautiful, novel, etc., there were a lot of biochemistry studies that had used that protocol, and some recent physical studies also that had used this protocol. So the protocol by itself, or the method of purifying the filament itself, uh, was based on the methods developed by other people. There was some novelty in that procedure as well, but I'll not go into the details of what the novelty was there in our method. And so here, what we have done is that we have added tracer particles. So these black dots are actually 200 nanometer polystyrene beads. And then the filament is anchored at one point on the surface, and we are watching it beat. So over many beat cycles, you can follow the motion of these beads and determine the fluid flow. So this is a high speed, high resolution movie, and if you watch it over many beat cycles, you will see the unidirectional flow. So from this kind of data, what can we get? So we can of course get the waveform, which will tell us about the elastic stresses that are acting in the system. And we can get the flow field, which will tell us the hydrodynamic forces. Now, in order to actually quantitatively compute all of these, I will need the material parameters. For example, what is the bending modulus of this object? Or what is the drag coefficient for this filament? And these parameters are well known in literature, consistent across diverse experimental probes. So, so here I'm showing you the digitized version of the waveform. And you can think of this waveform in terms of a power stroke and a recovery stroke. So from here, the recovery stroke begins, right? And this is an example, because we have this high time resolution, spatial resolution movie, we can measure time dependent flow field. So in the sense that at each time point in the beat cycle, we can associate a flow field with this filament. And here it's a power stroke, so the fluid is being pulled from here and taken here. So the important detail of this experiment is that because this is an in vitro assay, I can control the beat frequency with the ATP concentration, how much fuel I am adding. And there is a critical ATP concentration at which the spontaneous beating sets in. So there is no beating below a critical concentration, and above which you get oscillatory beating. And all our measurements were in this region of critical ATP concentration. And that is because we intentionally wanted to be in that regime so that I can throw away most of the microscopic details and keep it as a coarse grain model where I'm thinking of the cilia as a shearable active beam with planar motion. And I'm going to calculate stresses averaged over the cross section and use the information from the experimental data to determine the contribution of each term in the equation of motion. So I'm not going to, unlike other studies where you write down the equation of motion and then try to fit the waveform, I'm going to actually measure the magnitudes of each of the contributing stresses and then decide what should be the equation of motion. So the first thing that I need to do is to verify that the resistive force theory or the form of the viscous drag works. So because I have this slender deformable object, it's not clear whether I can simply write my viscous force as proportional to the local velocity of the filament. So because you can get these hydrodynamic coupling across different sections of the filament itself. And so in order to know whether the form of viscous drag is reasonable or not, we measured this flow field. So this is the flow field in part, at some point in the recovery stroke. And then we said, okay, the simplest model is resistive force theory, which says that the viscous force is proportional to the local velocity of the filament times the drag coefficient. And then I can theoretically calculate the flow field by saying that, I'm going to have a series of Stokes slits on this filament of strength Fv. And then I should convolute it with the Oseen tensor, which is the flow field for point force. And then because the filament is close to an O-slip wall, I need to use a different tensor, which is called the Lorentz-Blake tensor. So when I do that, and I know the local velocity of the filament from the waveform, and I know the drag coefficient from literature. So I substitute all of this information from the experiment here and get a theoretical computation of the flow field. And what we find is that there is a quantitative match between the two. And therefore, we were confident that resistive force theory is the correct path to model the fluid friction. Now I'm going to show you how we think about filament kinematics. So remember, we are modeling the cilia as an active shearable beam of a finite cross section A, and the best way to describe the conformation of the filament is to use a Fresnel array frame and associate a tangent normal and binormal with the center line. And you can, because this filament is bending, there is shear present in the system, and that shear is related to the tangent angle in this manner. So the shear displacement is filament diameter times delta theta. So now what I'm going to do is I'm going to plot the tangent angle. So basically this angle that's measured with respect to the horizontal as a function of, so as a chemograph. So this tangent angle across the contour of the filament for several beat periods. And so what you see is that you get this characteristic fingerprint texture, which is reminiscent of a traveling wave phenomena. So we can fit our experimentally measured tangent angle in this traveling wave format. And we have, see, the average shape of this filament is not flat, it's actually bent. And so this theta bar S is the average shape, which we are going to throw away because we are interested in the dynamic shape changes of the filament, and just going to keep the traveling waveform part. And now I'm going to write down the force and torque balance equations. So basically we are thinking of a cross section within the filament, and looking at all the forces that are acting on the cross section as well as on the surface, and some of those should be zero. So so this is the force balance equation, and you can easily integrate it. The only external force that this filament experiences is the viscous force, so you can integrate it to get the stress resultant that's acting on the cross section. Similarly, you can write a torque balance or a moment balance equation. Now, because we have planar beating, all of these terms are in the binormal direction. So you end up having a scalar force, scalar torque balance equation. Notice that you have this Fn term, which is the normal component of this capital F, that you can express in terms of the viscous force. So the viscous contribution is carried by this Fn term, and capital M is the moment resultant that's acting on the cross section, and small m is the shear moment due to the motors, and also due to the shear elasticity. So now what we need to do is to use constitutive equations into this general torque balance equation, and these constitutive equations will relate these the moments that are acting on the cross section with the measurable quantity tangent angle of our experiment. So the first moment that I should think about is the bending moment, which says that the moment is proportional to curvature, and curvature is related to tangent angle as the first phase derivative of the tangent angle. There is another contribution, which is the shear moment in the system. So this system has shear elasticity. So because remember these doublets and these Nexin links. So these are the passive cross links that resist shear, and therefore you have a shear moment per unit length, which again depends on the tangent angle. And then you have the active shear moment due to the molecular motors. And so you substitute these in your scalar torque balance equation, and you get this equation. So the first term is the contribution due to bending rigidity. The second term is due to the shear elasticity. The third term is the viscous dissipation. And the fourth term is the active moment. So now what I'm going to do is I'm going to non-dimensionalize it and then ask the contribution, measure the magnitude of each of these from the experiments, except the active moment. I don't know the active moment experimentally. The remaining terms I can measure experimentally. And so I do that. And this is how these chemographs look like. So the point to note here is that the scale bars. So what you notice is, so Gn is the hydrodynamic dissipation. And what you notice is that quantitatively, hydrodynamic dissipation is 2 orders of magnitude smaller than the elastic stresses in the system. Now remember, this is a continuously driven system, and it is absolutely essential that the dissipation competes with the elastic term in order for the system to have finite oscillations, in order for the system to be steady state. And so this was in my opinion, this was a clear-cut quantitative way to show that indeed you need internal viscosity for self-consistency. And now I'm going to invoke the most general contributions to for internal viscosity and show that how this inconsistency of dissipation being not in similar magnitude as the elastic stresses can be resolved. So we used the most general contribution to bending moment, which is to say that there is a bending friction that depends on rate of change of curvature. Similarly, on general principles, there is a term in the shear moment where the shear friction depends on the rate of change of shear displacement. And so here I really need these drag coefficients, right? The bending friction coefficient and the shear friction coefficient. The bending friction coefficient, I have used it for what the microtubule experiments found. And the shear friction coefficient, we use some back of the envelope estimates from the motor experiment, as well as based on passive Nexen link parameters, and both were similar. And so so now I'm going to, so these two plots are essentially showing the curvature friction and the bending friction. And so now what I'm going to do is, because I know that fluid friction is so much smaller than the internal friction and the elastic stresses, I'm going to drop it. So by adding these terms, now you can see that at least you have shear friction that is competing with the elastic stresses, and there is some contribution from the bending friction as well. So now I'm going to drop the fluid term and just retain the internal friction terms and have this as the final equation of motion. Once you have this equation of motion, one needs to establish whether this equation of motion actually permits oscillatory solutions. So to be able to do that, I need a constitutive model for the active moment. So now we are going to basically saying that theoretically whether this sort of formalism allows oscillations or not. And so we are going to use one of the simplest models of active moment, where you say it's first order in time, first order in space, zeroth order in space, and there is this B1 coefficient which decides the autonomous dynamics of M8, and the B3 coefficient which decides the feedback from the tangent angle of the filament. And so now I have these coupled equations of motion which I should solve and look for oscillatory solutions. So
Prerna, just interrupting you for
Correct.
one moment here. Just try to wrap it up in 5 more minutes, maybe.
Yeah.
Is
That
okay?
is the plan. Yes.
Okay.
I'm going to stop after this slide. Yeah.
Thanks.
So we solve the dispersion relation and look for oscillatory solutions in the B1, B3 space. And indeed, because we are doing a linear stability analysis, we can only see a transition between decaying and unstable oscillations. And the experimental beat frequency enables us to fix the values of B1 and B3. So, and what I wanted to show was that this decaying to unstable oscillation in the B1, B3 space does not really depend on the exact values of the bending friction coefficient and shear friction coefficient we use. Over a large range of these values, we continue to get oscillatory solutions. And I'll skip this slide in the interest of time. And I'll just stop here. Here, the conclusions of our study is that we have shown experimentally that resistive force theory is a valid form of hydrodynamic drag for beating cilium, that is beating close to the critical ATP concentration. We have direct experimental evidence that the hydrodynamic drag is negligible compared to the passive elastic stresses of cilia. And internal dissipation mechanisms are essential to have a self-consistent understanding of ciliary beating. So I'll stop here. If there are any questions, I can take. Oops, how do I? Oops.
Thanks, Prerna, for the very nice talk. Any questions from anyone?
Now I can sort of come here. Yeah.
Maybe I can ask a very basic question. I still find this idea of internal friction a bit, it's new and it's a bit counterintuitive. So I'm just
Right.
trying to understand if I got it right. Suppose if I mean, from your experiment with a single isolated flagella,
Yeah.
you could estimate quantitatively the internal friction. Now, instead, if I was dealing with the full cell,
Yes.
the Renharti, then I would expect a larger magnitude of internal friction, right? Because there are more nuts and screws inside the full cell body. Is that
Yes.
right intuition?
Yes. It's the right. I just wanted to do minor correction. I did not measure internal friction directly in my experiment. In my experiment, I measured external friction quantitatively, compared it with elastic stresses to conclude
That there is an internal
Yes.
friction required to fit that.
Yes,
Yeah,
yes.
that's what I meant by
Yeah.
estimating quantitatively.
Right,
Yeah.
right.
Okay. Yeah.
Yeah, and when you're thinking of filament on the cell, because the filament is now embedded in a basal body, like some kind of hook within the body, then that hook will also add a source of friction and so on. So you are right. In that sense, there will be other sources of internal friction as well. Yeah.
Right. Okay. Any other questions from anybody else?
I'm sorry, it was a bit technical. I
No,
tried
no,
to simplify it.
No, it was not so technical. It was okay. It was a good talk.
Yeah, thanks.
Any other question from anybody else? Any of the students? Okay, everybody is virtually silent.
Actually, I can make
yeah.
a minor point about this ATP thing.
Sure,
that you're
sure.
doing, right? So as maybe some of the students would have wondered that experimentally the fuel will get over at some point and then the filament will stop beating. So how are we taking care of the fuel consumption? So we have this ATP regeneration system, so some kind of coupled chemical reaction that is happening in the background that maintains a constant ATP concentration for like an hour. So we can make measurements. We are not worried about our fuel running out while we are making measurements.
Okay. Right.
Okay, if
Okay,
it
if
was
there are
crystal
no other
clear, then I can stop.
Yeah, if there are no other questions, let's thank Prerna once more. Thanks for being here, and thanks for this very nice presentation.
It was my pleasure to share my work. Thank you.
Sure.
Bye.
Thanks. Okay. Bye.